The measure of intelligence is the ability to change. Welcome all. Dr. Clark here again. And we're going to start discussing chemistry. I realize that many of you, just hearing the word, might be a little bit intimidated by chemistry. Seeing these, molecules, these dot patterns, etc., you might be intimidated by chemistry, but we're going to lay a baseline with chemistry. So when we start talking about things that are pertinent to biology today, like vaccination, like gene therapy, genetics, ecology, evolution. When we start talking about these themes, there's always a chemistry baseline to all these themes. And so I want you to learn a little bit of chemistry so you can learn a lot of biology. Okay? So that's where we're going to go in kind of this lecture. Just, again, laying down the baseline so you have kind of a basis to build a lot of these bigger topics and more pertinent topics to today's society as the current issues. Okay, so first off, let's talk basic chemistry. So all organisms on this planet, if they're considered a living organism, they're nothing more than a chemical machine. So they're built of atoms. Those atoms are linked together to form molecules. Those molecules are linked together to form macromolecules. Those macromolecules are linked together to form often organelles or some kind of system. Those systems, those organelles are linked together to form cells. Cells are linked together to form tissue. Tissues linked together to form organs. Organs are linked together to form a system, like the nervous system, or the digestive system, or the respiratory system. And then you're into whole organisms, and we'll progress whole organisms and their role in the environment. Okay? But we have to start at the basic building blocks of life, and that's atoms. So it's often said that chemistry is the science that roots all other sciences. And this is a true statement because chemistry often is studying the most minute, the most simple pieces of life. Those are the building blocks of life, like carbon-based compounds or atoms or water. Right? You have to know what the material is that organisms are built out of in order to understand biology or understand life itself. The opposite is not necessarily true. You don't have to know biology in order to study chemistry. And so you'll often see in college science classes, if you're going to have a science major and maybe you're majoring in biology, you'll have a biologist that's taking 4 courses in chemistry. But when you talk about maybe a chemist, they don't have to take a single course in biology. They can, but they could take any number of general science courses. They could take physics, they could take geology, environmental science. They don't have to take biology. But biology is different. And you'll see why once we start going, because it's that chemistry or those chemical machines. And that chemistry is paving the way for different processes to work. Most of you are fairly familiar with mass and matter, or at least you've heard of it before. So matter is anything that is comprised of a mass and takes up space. So anything that has a weight to it and occupies some form of space is considered matter. Now, that occupies some kind of space and mass is related to atoms. Everything that we know that is considered matter is made up of atoms. Okay, so all matter is made of atoms, or an atom. Okay. What are atoms? Atoms are the basic unit of structure. It's the most simple form of structure we know currently on the planet today. We're going to talk about two types of nuclei or nucleuses in this course. We're going to talk about a nucleus from an atom's perspective, or from a chemical or a chemistry perspective, and that would be the nucleus of the atom, which is comprised of protons, okay, and they have a positive charge to them, and neutrons, which have no charge to them. Okay, so that is a nucleus in the chemical form. Okay, so all atoms have a nucleus, which is comprised of protons and neutrons. We're going to talk about other nuclei or nucleus, okay, and those come at the cellular level. So quite a bit stepped up from the atoms. We have another nucleus, and the nucleus, the term just means basically center. It means, or control center. It's the center of something. So we're going to talk about atom nuclei, which is protons and neutrons, and then nuclei that occur in cells, which is where you would find DNA, RNA, ribosomes, nuclear envelope. Okay, that's the control center of a cell. We'll come back to the nucleus of cells later. Around the nuclei of an atom is another subatomic particle, and that is electrons, and they have a negative charge. Electrons will play a large role, a big role in future lectures, because electrons are often the source of energy that we're talking about when we're talking about the ability to do work. They can represent both potential energy and kinetic energy. We'll come back and talk about those topics in future lectures. But potential energy, in a nutshell, is stored energy. The stored energy comes in the form of electrons. When bonds or when chemicals are broken down, those electrons are allowed to migrate, move, and often that is what the kinetic energy is based on. Again, we'll cover energy in later lectures. I'm just, kind of laying the groundwork here for what electrons represent in biology. Now, a couple key components when we're talking about generalized chemistry, when we're talking about an atom. Remember, the nucleus is made of or comprised of protons and neutrons. The proton, or the number of protons, determines what element we're talking about. So if we have one proton, we're talking about hydrogen. If we change that proton and we add 5 more protons to the nucleus, now we're talking about carbon. So anytime you change the number of protons, you change what element you're dealing with. So the atomic number of an element is the number of protons in the nucleus. Again, if you change the number of protons, you're going to change the atomic number, which changes the element. So as long as you keep the same number of protons, the atomic number doesn't change, and you're dealing with the same element. Mass number is a little bit different. The mass of an element, or the mass number of an element, is how many neutrons and protons there are in the nucleus. So you add them together. So if we're saying the mass of, let's say the mass of carbon is 12, then when you would know, based on the atomic number, I just told you it was carbon, so you already know the atomic number is 6. It has 6 protons. Well, if the mass is 12, then you know how many neutrons it has. It has to have 6 neutrons. You can alter the mass number by altering the number of neutrons. So you could add a neutron to that carbon molecule, and now instead of having a mass number of 12, maybe you have a mass number of 13. Then this is still carbon, but now it's a different form of carbon, which we call carbon-13. It's an isotope. We'll come back to this in just a second. Now, like I said before, electrons are important to biological processes, biological pathways. The transfer of energy from one form to another is all based on electrons. And it's all based on the electron's behavior. Electrons get their behavior based on their position around the nucleus. So what occurs is, remember, electrons have a negative charge to them, and protons have a positive charge. So they're attracted to each other. This attraction has to do with the chemical behavior of the atom. If you have a tight attraction, the atom is likely not going to give up electrons readily. So often that atom won't react well, or it won't be quick to bond to other atoms. But if you have some electrons that are floating far away from the nucleus, maybe they're out there by themselves, and we'll talk about these orbitals that I'm talking about being out there, but maybe they're out there by themselves, and they're not really tied all that tight to the nucleus of that atom. In those cases, they might readily bind or readily bond with another atom to share that electron between two nuclei. We already talked about this. Electrons are associated with energy. We're definitely going to look at this again because majority of the processes that we're going to talk about in this course around energy is based on things like the ETS, or the electron transport system. Okay, the ETS, or electron transport system, is based on electrons being passed from one molecule to another and creating proton gradients. I know all these words, all this material sounds, confusing right now, but it won't be confusing when we cover it in those different sections. So we'll spend a little bit of time talking about photosynthesis, a little bit of time talking about cellular respiration, so you understand where these electrons are moving, where this material, where this energy is coming from, and then how is that energy converted from one organism to the next. Now, again, I talked about potential energy and kinetic energy. Okay. Electrons naturally have potential energy or stored energy. Okay. And so that stored energy can be released when the electron changes its position. It can change its position by having a bond that's between one nucleus and that electron being broken or removed, so its association with that nucleus is now gone. That changes the position of that electron and therefore can give off energy or can require energy in order to change that position. If you're moving an electron closer to a nucleus, the energy that it takes to hold that electron in place is less when it's closer to the nucleus, so energy is released. And we'll look at some examples of this. Around the nucleus, you have electron shells or electron orbitals. And so these are the space or the area in which you're most likely going to find an electron. So it is going to be floating around the nucleus, associated with a nucleus, and it's going to be floating around it in these electron shells, okay, otherwise known as electron orbitals. The space is actually an orbital, and so it orbits the nucleus. Now, each electron shell has a specific number of orbitals. These orbitals can either be filled by electrons or not filled by electrons. But orbitals that are devoid or don't have any electrons often will readily bind to something to fill that orbital. So if you're missing an electron to complete your orbital, and you are put into solution with something that has an electron that you could share with another molecule. So let's say we take carbon, which is missing 4 electrons. It needs 4 electrons to complete all of its orbitals. Let's say we take that carbon and we put it in solution with something that carbon molecule can bind to. So maybe we put it in solution with water. Right? And these hydrogens that are in water, they can bind up to that carbon molecule. And it can fill those orbitals with the electrons that are orbiting each hydrogen molecule. You have a single electron that's orbiting it. Okay? So they can share those electrons, and that's going to fill those electron shells, and it's going to make a more stable molecule. So in general, in a general rule of thumb, atoms that have incomplete electron orbitals, so they're not completely full with electrons, they tend to react more often. Okay? Atoms that have complete electron orbitals, they tend to be most stable when they're not bound with something else. Okay? So if that atom has an incomplete electron orbital, depending on how incomplete it is, that atom may gain electrons, it may lose electrons, it may share electrons. It depends on what that atom is coming in contact with. What's the other atom that atom is coming in contact with? That sharing of electrons is the basis of what we call bonding. And we'll cover a couple types of bonds as we progress. Now, before we get into bonds, though, let's talk about that electron behavior, or that electron potential energy. Okay, so here you can see these energy levels, and you can think of these energy levels here in this picture as the orbitals around that nucleus. Okay. If an electron is out here on the outermost orbital and it is moving closer to the nucleus, you don't need as much energy to hold that electron in place or in position the closer it gets to the nucleus. So when that electron moves closer to the nucleus, energy is released. The opposite is true. If the electron is in a close position to the nucleus, to get the electron to move away from the nucleus, you have to apply energy. Okay, so energy is released to move closer to the nucleus. Energy has to be applied to move further away. You can think of it just like two magnets. You have a positive magnet and a negative magnet. And you're moving that negative magnet closer to the positive magnet. At a certain point, you can let go of that magnet and it will automatically, okay, due to the attraction, go to the positive end. That movement is going to release energy. Okay? That energy is being released. You don't have to apply energy. It automatically releases energy to make that connection. Okay, but then let's say you have a magnet, negative and positive, and they're close together or even stuck together, okay, and you're trying to separate them. You're trying to move that negative away from the positive. Okay? You have to supply energy to a point. Okay, once you supply energy to a point, you've kind of broke that connection, and that electron can now move away from the nucleus. But you have to supply energy. This is important because we'll start looking at molecules and their attraction to other molecules, and then the importance of molecules in the form of stored energy or potential energy. So like I said before, moving electrons further out from the nucleus requires energy. Moving them closer to the nucleus releases energy. All right, let's talk about a little bit about ions and isotopes. Okay, over here you can see this woman here. She's probably the most famous female scientist. Okay, that's Marie Curie. She's the only female to ever win the Nobel Prize twice. Okay. She won the Nobel Prize in chemistry and physics. And one of her Nobel Prizes was basically the discovery of isotopes, right? Or the half-life of isotopes. And so we'll cover her discovery and the role that she played in the discovery of chemical isotopes and how we use isotopes in biology today. Before we get to that, we'll talk a little bit about ions. Okay, ions are a form of an atom. And the form of an atom when it either gains or loses one or more electrons, it is called an ion. Okay? And the reason why it's called an ion is because it has some ionic charge often. So if you lose electrons, then the atom may become positively charged. If you're gaining electrons, then that atom may become negatively charged. Okay? And so this is how you get an ionic form of an atom, or a different form of an atom based on the loss or gain of electrons. Isotopes. Okay? Isotopes are atoms that have the same number of protons but a different number of neutrons. So back to that carbon example. When I said carbon has 6 protons and carbon 12, or the mass number of 12, it has 6 neutrons. Technically, that is considered also an isotope of carbon, but that's the normal form of carbon. That's the most abundant form of carbon. 99% of all carbon is carbon 12. It has an equal number of protons and neutrons in its nucleus. You change the number of neutrons, say you add 1, now it's carbon 13. You add 2, now it's carbon 14. Okay? And so these iterations of these different elements occur often. So there's lots of different isotopes of lots of different elements that occur naturally. Okay? In some cases, there's a higher percent of that isotope than in other cases. So in the case of, say, something like carbon 14 and carbon 13, and they only represent together maybe 1% of all the carbon on the planet. Okay, but in other elements, maybe the isotope represents 5% or something along those lines. Okay, so elements will have a different mixture of isotopes in nature. So let's jump to this isotope. And this is Marie Curie's work. She was looking at isotopes, and she was looking at what we call the half-life of isotopes. How long do isotopes occur in that unequal form? So with an abnormal or non-equal number of neutrons, how long do they occur in that form? So if we look at carbon 12, like I said before, about 99% of all carbon is carbon 12, which means it has 6 protons, 6 neutrons, and 6 electrons. Okay, again, if you change the number of electrons, you make an ion. Okay, but if you change the number of neutrons, you make an isotope. In this case, I'm just showing you the isotopic reaction. So if I add a neutron to carbon 12, it makes it carbon 13. It changes the mass number, it changes the mass number, but the atomic number is the same. If I add another neutron, so now there's 8 neutrons, now it's carbon 14. The electrons haven't changed, the protons haven't changed. We're just changing the number of neutrons. That's the isotope or isotopic reaction for carbon. Now, Madame Curie discovered a way to figure out how long does carbon 14, how long does it stay in that form with those extra neutrons? And then what happens to it? Do these neutrons go away? Do 2 protons eventually get added to this? And if it does, then it's no longer carbon. It's now a different element. Is it nitrogen? Okay, we're going to examine what happens to this when we talk about radioisotopes and isotope dating, okay? But how long something like this occurs in the system is called the half-life. And we'll come back to that in a little bit. Okay? So Madame Curie came up with the idea of what is known now as radioactive decay, or the isotopic period. Okay, so how long that isotope stays in the system. So again, isotopes themselves, by not having the same number of neutrons and protons in the nucleus, they'll often be unstable. And they'll tend to break up, or they'll tend to be radioactive or have a different chemical behavior to that molecule. Okay? So in some cases, it could be giving off energy. Okay, they're not stable, so they're reacting with other things. Okay? In other cases, they might be perfectly stable, but they decay over time. Now, in today's society, when we're talking about biological sciences, we can use this method that Madame Curie first described of radioactive decay. We can use it in quite a few different forms. We can use it in medicine, and we do so quite often, and that is to use it as tracers, right, or detection devices. You can use things like a radioactive form of glucose. This is often used. Okay? A radioactive form of glucose. You can use it and inject it into the body of a human, right? Or for that matter, probably any mammal you can do this with. And you can trace where that glucose is going. So it's going to send off electrical pulses. This material is going to send off electrical pulses. We can use a scanner that picks up those electrical pulses, and those pulses are generated often wherever that glucose molecule resides, wherever it's going, and it stays in that spot. It's going to send off electrical pulses. We can scan it and we can see those electrical pulses. We can see that tracer where it went and where those tracers are now established or sitting. Well, it just happens to be that if you use glucose, glucose is going to regions where there's a lot of cell growth, a lot of cell regeneration, like cancer cells, tumors. Okay? They're regenerating cells really quickly. Right? And you can see on a scan, then you can see these glowing areas or areas where there's a lot of energy being radiated off, and that would indicate that there's a tumor or there's lots of cell growth in that region. Okay? We'll look at some more examples of this. Okay? Dating fossils. We can use something that lasts a lot longer. So the ones that we use for medicine, maybe their half-life is a matter of hours, or at most maybe a day. So that means that material is going to deteriorate very quickly. So you're going to have that radioactive material. It goes in the body. It generates these electrical pulses, and then it's gone. It's decayed over time, and it's harmless to the human. When we're dating fossils or dating objects from the past, we want things that last longer, that have a half-life or a period of decay, a known period of decay again. Okay, we need to know what the decay is, but we want that decay period to be longer so then we can date how much of that material is left in that object after a given amount of time in the ground, or wherever that object is found. Okay, so isotopes, like I was talking about before, here you can see different scans. Okay, sometimes they're like blot scans, sometimes they are these more colorful energetic scans. Okay, CT scans. But at any rate, you're using an isotope that is very short, has a very short half-life, so it decays very fastly. It's not going to harm the individual that you put it in. Okay, they can drink it sometimes. Sometimes it's injected. But at any rate, it's going to go through the system and it's going to interact with often areas of extreme cell growth. There's other ways that we're starting to use isotopes also, where we can label biochemical pathways. So maybe we're interested in what position or where certain things get absorbed in the body. Maybe we're designing new drugs and we want to make sure that this drug is being absorbed where it needs to be absorbed. So you can trace that. You can label that drug, and as it enters, it sends off a radioactive label, and you can track where it is going in the body. So maybe at first, after digestion in the stomach region, maybe it enters the bloodstream, it gets incorporated in the liver, maybe it stays in the liver, or maybe it goes into the bloodstream and it goes straight to the brain. Where are these molecules going? And we can use isotopes for that kind of tracing of molecules throughout the body. And we're just starting to get good at this. Okay? We're starting to be able to trace DNA and starting to be able to trace individual genes to see what happens to genes. Now, there was a researcher, her name is Barbara McClintock, who in the 40s, she said that genes could jump around on a chromosome. People thought she was nuts. Okay? She called them jumping genes. But she said genes were moving from one spot on a chromosome to another spot on a chromosome. Okay? They weren't always in the same position. Well, we now know, because we can use tracers and other technology, we now know she wasn't crazy. She was exactly right. Okay? We now know that indeed genes can move on a chromosome. And we'll come back and we'll look at what is the implications of that movement, okay, on the position of a chromosome. All right, and then dating fossils. Fossils are fairly important, for geology, but it's also important for evolution and biology, and even fossilized humans, so archaeology or fossilized remains of other things, right? Or often we use dating when it's not even fossilized, it's just old material, so maybe even in the form of theology, and we're looking at religious doctrine and history, and we're looking at the history of certain maybe certain types of clothing, or maybe certain types of writing and things like that. We can use isotopes, okay, to date that material. So the way isotopes work is they have a radioactive decay, or they have a half-life which is constant. Okay, it's a known number. If you examine material that has been, deposited underground, maybe as a fossil, or deposited in a tomb or something like that, and you examine that material, there will be a certain percent of decay of the molecule that you're looking at. If it's a material that is human-based, so kind of within the last 30,000 years, then carbon-14 is often used. If it's something that's prehistoric, like a dinosaur, maybe you're looking at a dinosaur and it's 300 million years old or something like that, you'll use something maybe that has a much longer half-life, like potassium or something along those lines, which the half-life is much longer than the half-life of carbon-14. Okay. The older the fossil, the greater the decay of the given material, the given isotope that you're interested in. So here's an example, okay? So we know that organisms, while they're alive, okay, so like this rabbit, while it's alive, it is consuming plant material. That plant material will have a certain ratio of carbon-14 to carbon-12. We know this because we can test, what that ratio is. So if you can identify the plant that organism would consume, you then can identify the ratio of carbon-14 to carbon-12. That organism would have consumed it. You know the ratio of it. That organism dies. You know what the starting ratio would be. And in the case of carbon-14, we know that every 5,730 years, carbon-14 would be decayed in half, or that's the half-life of carbon-14. So let's say you have 1 gram of carbon-14 in the skeleton of an organism, or that's what you would, based on what they had when they died, is 1 gram maybe for every, 100 grams of carbon-12, maybe you have 1 gram of carbon-14. Well, after 5,730 years, you should have half a gram of carbon-14. After another 5,730 years, you should have a 1/4 gram of carbon-14. After another, and it goes on and on and on until really it's not detectable. Okay, so you would be able to then detect what that organism, or how long ago that organism had a solid amount, or the original amount of carbon-14. You just back calculate it. And so if you decide that it had 2 half-lives of carbon-14, you would then say that this organism was alive approximately 11,460 years. It gets a lot more complicated than that, but this is a way that you can do it by hand. We now have machines that calculate it by not just full, half, quarter, eighth, 16th, but even much more minute numbers, so you can get a better accuracy than every 5,730 year kind of situation. But one key component to this is that carbon-14 is not going away. This goes along with the age-old saying, the first law of thermodynamics is energy is not created nor destroyed. Well, in fact, nothing is created nor destroyed. It just changes form. So carbon-14 is not being destroyed over time. It is being altered and changing into nitrogen-14. So over time, that carbon-14 is decaying and it's changing into nitrogen-14. Okay? All right, so we'll come back and we'll look at ions and isotopes. Okay, we'll look a little bit more at some isotope uses in medicine, and uses when we talk about evolution, and how we know certain organisms are a certain age, and how we know the planet is a certain age. Okay? And then we're going to look at ions when we start talking about energy and the transfer of energy from one source to another. Okay? But next time, we're going to continue with chemistry. We're going to talk about some molecules. We're going to talk about pH. We're going to talk about bonds and how atoms link together next time.